The next and the last speech of this session will be given by Ling Peng from Aix-Marseille University in France, and she is going to talk about the modular and adaptive self-assembling dendrimers for nanomedicine. The floor is yours.
Thank you. Thank you very much, and thank you for Bert for the organization for this invitation. I'm very happy to share with you some of our recent results about self-assembly dendrimers for biomedical applications. In particular, we talk about modular and adaptive, because I think yesterday you all heard about Jean-Marie's talk, a supramolecular system to adaptive system. So before I come to the, I think in the audience yesterday, Mariana already told about you what is dendrimers, and also dendrimers, actually dendrimer is not according to the strict scientific definition, dendrimer is not a complex system, because dendrimer is a very precise material that have the structural component of core, branch unit at the terminals. And also dendrimers, you have the well-defined structure, and they have unique multivalence, and they have the inner cavities to encapsulate the drug for drug delivery, and on the surface you can conjugate the drug for drug delivery. So actually the ideal and the precise materials for drug delivery. But unfortunately, when we synthesize dendrimers, you can look at this dendrimers is tree-like molecules according to the Greek word. And unfortunately, when we synthesize the dendrimers at the higher generations, sometimes we always inevitably we have the structural defect. So then it makes dendrimers complex systems. And here it's just to show you that the dendrimers we synthesize in our lab, it looks like a tree, really tree-like molecules, composed of the core, branch unit, and the terminal groups. And this is to show you if the dendrimer is a precise, well-defined structures, how can we make the nanosystems very simple, symmetric system, not complex at all if all of these things happens in the ideal situation. So actually, as Marianne told you yesterday, that it is actually is not very it's not a perfect system. So what we are thinking about is that we try to use the small amphiphilic dendrimers which can self-assemble into supramolecular dendrimer nanosystems. And we hope that this self-assembling long-covalent supramolecular dendrimer nanosystems can mimic the well-defined covalently constructed dendrimers for the size, shape, and also for functional deliveries for drug delivery. So that's what we are trying to do in our group. And since these amphiphilic dendrimers, they form the nanomicelles because of the dendritic structures, they can form easily nanomicelles. So we can use the hydrophobic interiors to encapsulate hydrophobic drug. And also we can make the surface positively charged in order to deliver the negatively charged nucleic acid. And also we can modify chemical modification on the surface for drug delivery or for imaging agent delivery. That's what I'm going to show you at the moment. And also if we compare the amphiphilic dendrimers from the nanomicelles, it has unique difference from the conventional lipid nanomicelles and the polymer nanomicelles for drug delivery. Because for the lipid and the polymer nanomicelles, they have a dense core, hydrophobic core, so the loading will not be very high. If you look at the amphiphilic dendrimers, which form the nanomicelles, the core, hydrophobic core, have a lot of space. So if you have a lot of hydrophobic space, you can load it with a lot of drug. Then you can have a high drug loading. And I remember yesterday we talked about often the high drug loading have the better effect. So this is the unique advantages if we can use amphiphilic dendrimers to form the dendrimer nanomicelles for drug delivery. And indeed, we can achieve very high drug loading more than 40%. And also we can modulate the size because we can change the hydrophobic part and the hydrophilic part to get a different size. So according to us, because we want the nanomicelles, that nanosystem to be small, so actually we try to control the nanomicelle size, which is around 15 nanometers for the dendrimer nanomicelle. And this size is very difficult to be achieved using polymers. And here is to show you that the dendrimers, we synthesize amphiphilic dendrimers and they form nanomicelles. And in the upper panel is the empty dendrimer nanomicelle. In the down panel is that the dendrimers is loaded with drug. Here is the doxorubicin anti-cancer drug. And you can see the nano size is around 10 to 12 nanometers. And the drug is encapsulated inside. And these nanoparticles can accumulate into the tumors via EPR effect. And also they can penetrate very deep into the tumor models. Here is the 3D, I don't have the 3D tumor models. And also these nanomicelles can release the drug at acidic conditions. They have the acid responsive drug release because usually the tumor micro environment has these acidic conditions. And most importantly, when we dissect the tumor tissues, we discovered that the dendrimers, that the tumors, the dendrimer nanomicelles is covered, is spread all over the tumor in vivo in the tumor tissues. So we ask us the question, why our dendrimers nanomicelles can go deep into the tumor tissues and spread all the tumor tissues so they can have effective anti-cancer activities. To answer this question, and we try to think about what is the mechanism. And then we think about maybe that this our dendrimer nanomicelles, once they were taken up by the cancer nanocells, and the cancer cells will generate extracellular vesicles. And these extracellular vesicles can be, our nanoparticles can be repackaged in situ in the cell, in tumor tissues, in cancer cells to generate the extracellular vesicles. And these extracellular vesicles can carry the nano drug to further to deliver to the other cells for intracellular delivery. And this, because of this delivery process, they can have deep penetration. And also because extracellular vesicles was generated by cell inside the tumor tissues, inside the microenvironment. So we know that the tumor microenvironment is heterogeneous and dynamic evolved. So our dendrimer nanoparticles can be delivered by the extracellular vesicles according to the heterogeneous microenvironment and the dynamic features for the delivery. And indeed, we can isolate the extracellular vesicles, and these extracellular vesicles that bear the extracellular, they have the size of the extracellular vesicles, they have the biomarks of the extracellular vesicles, and we can show also that the dendrimers can be co-loaded in the extracellular vesicles. And also the delivery and the penetration of our dendrimers into the tumor spheroid, organoid models can be inhibited by the extracellular vesicle inhibitors, and also by the inhibitors which inhibit the uptake of extracellular vesicles. So with all of this data, we convinced that this extracellular in situ tumor-generated extracellular vesicles plays a very important role for our nanoparticles spread inside the tumors, homogeneously go over, and also can overcome the heterogeneous and the dynamic evolution. And here is to show you that the results using our dendrimer nanomicelles, we can deliver the different anti-cancer drugs, doxorubicin, paclitaxel, and rapamycin, and they can overcome the anti-cancer drug toxicities. And even here is also to show you that the rapamycin can generate metastasis, and we can, using our Dendrimer nanomicelles, we can inhibit the metastasis generation. So this is to show that using our self-assembly Dendrimer nanomicelles, we can deliver the hydrophobic drug. At the same time, if we can make our amphiphilic Dendrimers positively charged on the surface, we can also deliver the negatively charged nucleic acid. And here is the results. What we show you is that this amphiphilic Dendrimer, the self-assemble into about 10 nanometers nanoparticles, and then when they interact with nucleic acid, they form around 30 or 50 nanometers nanoparticles, and then they uptake into the cells and they release into the cytosol for gene silence. Here is the example to show you the gene silence at the messenger RNA level and at the protein level, and with anti-cancer activities, because we use the siRNA to target the oncogene MYC in these examples. And we show that it's very important to have the amphiphilicity, because if we have the small Dendrimers without hydrophobic part or with a hydrophilic part, there's no activities. And also we demonstrated that this because of the amphiphilic Dendrimers is a hybrid of lipid and the Dendrimers, so it can benefit the endosome-scape advantages of lipid vectors endosome-scape, and also the Dendrimer nanovectors endosome-scape by proton-spongy effect. And most importantly, that we have developed the amphiphilic Dendrimers which can deliver the nucleic acid into primary immune cells, because primary immune cells is very difficult to deliver for the drug delivery, because they are very sensitive to exterior material, to foreign materials. Here's to show you that using our Dendrimers, we can deliver to primary T cell, primary natural killer cell, and also to primary monocyte and the microglia. And importantly is that we can make the Dendrimers have the conjugate, and which can do the click chemistries with antibody, and we can use the antibody which against the PD-L1 ligand of the cancer cell for targeted delivery. And here is to show you that using Dendrimers which build the functional groups for click chemistry, which click to the antibody for the siRNA delivery into the tumor cell, and at the same time they block PD-1 and the PD-L1 immunoaxis have the immunoactivities, so they can have the dual activities for anti-cancer activity, for anti-cancer effect. And also we try to develop Dendrimers which can be responsive to cancer cell stimuli, because we know that cancer cells have a lot of have a very high reactive oxygen species. So if we incorporate reactive oxygen species sensitive groups within our Dendrimers, so when the Dendrimers deliver the nucleic acid into the cells, they will be degraded by the high level of reactive oxygen species, and then they release nucleic acid. So in this way, we can also achieve specific cell-specific targeting to the high level of reactive oxygen species. And the most interesting that we synthesized a different size, different generation of the Dendrimers, and we discovered that the small amphiphilic Dendrimers, they are very effective for small interfering RNA deliveries, while the higher generation of the amphiphilic Dendrimers, they are more effective for the large nucleic acid, large DNA delivery, DNA transfection. And here we show you that using plasmid P51 and also siRNA against AKT2, we can achieve effective anti-cancer activities in the metastasis cancer models. And we compared our Dendrimers with the commercial vectors, jet lipofectamine and the jet PEI. I didn't show that. And also the lipid which showed by yesterday MC3 for specific for siRNA delivery. And you can see that with our Dendrimers, we can achieve even better siRNA delivery and the anti-cancer activities than the MC3 LNP nanoparticles to inhibit the metastasis. So I think my time is running, so I will finally talk about that using our Dendrimers, we can also decorate and load it with the bioimaging agent for bioimaging, for tumor imaging. So usually we have the imaging agent, we talk about the fluorescence imaging, and most fluorescence imaging agents are hydrophobic. So for hydrophobic imaging agent, we put that inside the nanomicelle, and for the hydrophilic imaging agent, we put them on the surface. So this is what we have done with on the upper panel is for the fluorescence imaging, and on the down panel is for the radio imaging, PET and the SPECT imaging. And here is to show you that we conjugate the chelators on the surface of the Dendrimers. We can chelate the indium for SPECT imaging, and we can see that the tumors is in the red circle, but also we have very high, heavy uptake in the liver. So to reduce the liver uptake, we try to change the chelators. And as you can see, when we change the chelators from DOTA to NOTA, we can significantly reduce the liver uptake. And also, we can chelate gallium for PET imaging instead of SPECT imaging. And with the galliums, we can see that we can detect the pancreatic cancers, which is often not detected by FDG, which is the clinical reference for PET imaging of tumors. And using our Dendrimer nano systems, we can see it very clearly for pancreatic cancer detection, while with the clinic reference, we didn't see any signals, and also the small molecules. And finally, we can also chelate the gadolinium on the surface for MRI imaging. And here is to show you that using gadolinium, we can do the MRI imaging and fluorescence imaging. So I will stop here. So here's our group. We have a very small group, and right now we are hiring for PhD and postdoc if you are interested. And here is all our long-time collaborations for cancer, for imaging, for Dendrimer characterization, and for different cancer models and the pharmacokinetics. Here is the financial support, and finally, thank you for your attention.